Okay, we'll just get started for the day. Hello everyone, welcome to week 13 of our molecular biology PMRF-NPTEL sessions, and today we are going to be doing our last extra session on molecular biology. This week we are going to go through some of the key concepts we have learned throughout the course. So there are going to be multiple different questions from the entire course, and I would recommend everyone to try and solve it together with me so that we get an understanding of how well prepared we are for the final NPTEL exam and how much we have learned from the entire course through till date. So a quick introduction for myself. My name is Anuvab Chatterjee. I'm a PMRF fellow at IIT Kanpur and I'm taking this course. Today I'm going to show you questions from all throughout the course, and uh, you know the same thing that we have been doing till now. I will ask you to try and solve it, and then we'll try and solve it together, getting a better understanding of whatever the concepts behind the questions are, and you can treat this as a recap for the entire course that we have been doing till now. So the first question that we have today is, in prokaryotes, apart from the single chromosome, some bacteria have extra chromosomal DNA which is known as plasmids, mitochondria, ribosomes, or endoplasmic reticulum. Developer says it is A. Chanda also says it is A, plasmids. So let us look at it. Both of you are correct. Plasmids are the prokaryotic organisms frequently carry one or more smaller independent extra chromosomal DNA called plasmids, and these are extra chromosomes, meaning they are not part of the genomic DNA. They are not essential for the bacteria to survive, but they give particular resistances to antibiotics or could play a role in and help the bacteria grow via other factors, and they are accessory DNA molecules. These are small circular DNA molecules that have the ability to self-replicate. And unlike larger chromosomal DNA, plasmids typically are not essential for bacterial growth. Why are they important? They give bacteria various advantages such as antibiotic resistance, herbicide resistances, or even transfer information between one bacteria to the other. And in addition, unlike chromosomal DNA, plasmids are often present in many copies per cell. So plasmids can be high copy number or low copy number depending on the ori site or the origin of replication or how efficient it is. And if it is very efficient, then you will have a large number of copies throughout the bacterial cell, and they are all uh, transcribing and translating genes that are present on them. So this we have seen how a plasmid looks at like. Just look at it again. So plasmids have an origin of replication. Here is where the replication will start, and this is autonomously replicating. So plasmids can replicate independent of the genomic DNA. It has an antibiotic resistance gene, which confers antibiotic resistance to the bacteria. It can also have a selectable marker. So this plasmid that we are looking at, it is a cloning plasmid that is used in molecular biology, and this has different things which help us uh, do molecular biology on the plasmids. So once a plasmid is present in a bacteria, you need a selectable marker to verify that the bacteria actually has the plasmid. Then it has a promoter enhancer that starts transcribing your gene of interest, which is cloned in the multiple cloning site of the plasmid. So this is where you will put your gene, and then you have primer sites that will help you do PCRs and check if your gene of interest is present in the plasmid. As we have seen, genomic DNA is separate from plasmids, and obviously this is much more smaller than genomic DNA. It is just not to scale, but and there will be a multiple number of plasmids throughout the bacteria. So in prokaryotes, apart from single chromosomes, some bacteria have extra chromosomal DNA called plasmids. Next, in plasmid isolation, the purpose of alkaline lysis is blank: to enhance the binding of plasmid DNA to the spin column, to denature chromosomal DNA while keeping plasmid DNA intact, to selectively denature chromosomal and plasmid DNA, or to digest RNA molecules. Uh,
Yes. Okay. So we have four options. We can put in whatever you think is the correct option in the chat. In alkaline lysis, what is the purpose of alkaline lysis and plasmid isolation? You can put in what you think is the correct option in the chat. Chandra says it is B, to denature chromosomal DNA while keeping plasmid DNA intact. Anyone else? The Palisade also says it is B, to denature chromosomal DNA while keeping plasmid DNA intact. Let us look at it. So we have gone through isolation of plasmids. We will go through it again today. So how do you isolate plasmids? You first grow the bacteria that has the plasmid, and then the first step is Using a solution of glucose to hydrochloride and EDTA. So this is nominally known as solution 1, in which you will resuspend the pelleted bacteria. So once you have a culture, you generally take the culture, if it is say 20 ml culture, you pellet it down into a falcon, which is and falcon 50 ml falcon, you can pellet it down, and you have a cell pellet. The cell pellet has dead bacteria in it, but the bacterial cell walls are not ruptured. So what you want to do is that you want to resuspend them back into a solution without rupturing the cell walls. So that is what you use the solution 1 for, which is an isotonic solution. So this will not, so the So the concentration of things inside the bacteria as well as the outside solution is same or similar, so that the bacterial cell wall doesn't rupture because of osmosis of water into the cell. So this step is a resuspension of the bacteria back into solution using a solution of glucose, hydrochloride, and EDTA. Glucose is for maintaining the iso isotonicity of the solution. Trihydrochloride is for maintaining pH, and EDTA prevents any DNA damage by chelating DNA enzyme cofactors such as magnesium. The next step is the alkaline lysis. In this step, we use a solution made of NaOH and SDS. Both of them together, what they do is that NaOH punctures holes into the cell wall, and SDS enters the cell and denatures everything. So in SDS is a detergent which has a organic part and a polar part. The polar part is extremely negatively charged, so it sits on any kind of organic molecules present inside, so lipids, proteins, even DNA, and then it helps in denaturing them. So they just unfold and then they come out of solution. So you will have a huge precipitate that forms once we put in the alkaline lysis solution. Or rather, not a precipitate, but rather the due to the change in the pH, it will become cloudy in nature. You will still not see a precipitate till you add the renaturation third solution, which contains potassium acetate and glacial acetic acid. When you add this, the pH comes back to normal, but any organic compounds such as proteins, lipids that were denatured, they come out of solution. So they will form a precipitate. But any small polar groups, such as your nucleic acids, they have the ability to actually come back into solution and will be present in the aqueous layer. And then then you centrifuge the whole thing, and then you will have a precipitate, which will be your cell debris, your lipids, your proteins that were able to be denatured, and your aqueous solution will have your DNA and nucleic acid. So in this step, small DNA such as plasmids renature back quickly, whereas chromosomal DNA remains denatured because chromosomal DNA is huge. Another thing that happens is that plasmid DNA, because it is small in nature, it can rehybridize back pretty efficiently. So it will rehybridize back into the small circular form that it denatured from. And thus it will easily be able to come back to solution. But genomic DNA is very large. So the renaturation is not efficient. So there will be huge loops of the genomic DNA which will be exposed. So the organic bases will be exposed to the polar solution, and DNA is not like that, and the entire thing will denature and be present in the precipitate. So you will not have any contamination of genomic DNA of the bacteria in your plasmid suspension. And then you do you once you take the so once you have centrifuged the solution after adding solution 1 to 3, you will have a pellet, which will be the cell debris, the genomic DNA, the lipids, and the proteins, and the aqueous solution containing polar groups that were small enough to renature back. Now you want to purify it even further. So some proteins, some lipids which are small enough will still be present in this aqueous solution. You add a mixture of phenol, chloroform, and isoamyl alcohol at this step. Phenol and chloroform, they are so phenol is, we know, a polar compound, and chloroform is organic in nature, but together they actually form a bottom layer which captures all the lipids as well as proteins, which have any organic group. So any organic groups will preferentially dissolve in phenol alcohol by phenol chloroform rather than stay in the aqueous layer. So everything that the protein or lipids that are still present in the solution will go down into this layer, and the aqueous layer will contain your polar groups such as your plasmid. And there will be a cloudy small layer of isomer alcohol when you add this solution to your tube, and that isomer alcohol is an ambivalic compound which has again organic tail as well as a polar groups added to it. So any proteins which are ambivalent in nature, which have both the organic group as well as a polar group, will actually get stuck in this isoamyl alcohol layer. Then you again centrifuge it very hard, and you will see three layers forming: the top aqueous layer containing your polar plasmids, the middle layer containing proteins which are ambivalent in nature, and the bottom layer containing any organic proteins, lipids, and anything else. And you just take the upper layer out, and that contains mostly contains your plasmid, but it will also contain salts. Salts are highly polar in nature, and they are actually still concentrated in this aqueous solution. In this step, you finally put in 100% alcohol. 100% alcohol lowers the solubility of the things inside this aqueous solution, and DNA, especially DNA, which is a big group which still has non-polar groups in non-polar parts in it, such as the organic bases, cannot stay soluble in the resultant solution after you add 100% alcohol. And when you precipitate it very hard, then you will see another pellet, but this time this pellet will have your plasmid DNA, and the aqueous solution will contain salts. You remove the salts and then you re-elute your plasmid, and finally you have isolated your plasmid of interest. To In plasmid isolation, the purpose of alkaline lysis is to denature chromosomal DNA while keeping plasmid DNA intact. Next, blank is used to lyse bacterial cells in plasmid isolation. The options are tris hydrochloride, sodium decyl sulfate, which is SDS, ethanol, or agarose. Okay, Shivakumar, Debadrita, and Chandra all say B, SDS. Let's just look at it. We have seen that in the solution 2 of alkaline lysis, you use a mixture of NaOH and SDS. SDS is what you use to lyse the cell. So lysis is essentially opening big holes in the cell wall so that the cell ruptures. But this is a controlled lysis of the cells. You do not rupture and then everything goes out, and then you have problems separating your DNA from everything else that the cell had. In this alkaline lysis, there are huge holes that form in the cell wall, but the entire cell is not ruptured open. So, and this is the step in which you have to be very careful because, as we have said, plasmid DNA as well as genomic DNA both have the property, because both of them are nucleic acid, to come to become part of the aqueous layer when you add renaturation solution. But the only thing preventing the genomic DNA from contaminating your plasmid DNA in the aqueous solution is the huge size of the genomic DNA, and the fact that in bacteria the genomic DNA is bound to the cell wall. So you have to be very careful in this lysis step that you do not shake the tube vigorously or vortex the tube, because when you do that, then the genomic DNA will shear. Genomic DNA will shear. That is, it will break apart into smaller and smaller pieces, and these smaller pieces will be easily able to contaminate your aqueous layer. And then you will not only have your plasmid, but a lot of other contaminating DNA that will do a lot of problems downstream. But to answer this question, blank is used to lyse bacterial cells in plasmid isolation. The answer is sodium dodecyl sulfate, or HDS. Next, low blood glucose level causes blank. dephosphorylation of pyruvate kinase, activation of glycolysis, increased insulin secretion, or phosphorylation of pyruvate kinase. Okay, so Shivakumar, Devadrita, and Chanda all said D, phosphorylation of pyruvate kinase. So let us look at it. So this part, this question is directly from glycolysis and cellular metabolisms. So let us quickly go through this. So we have gone through glycolysis. It is the major pathway in which glucose is broken down and you extract ATP and produce energy for the cell. So there are 10 different steps in glycolysis. The first is glucose gets charged to glucose 6-phosphate. This not only is a step towards the final breakdown of glucose, but also prevents the negatively charged glucose 6-phosphate from escaping the cell. So once glucose 6-phosphate is produced, then the glucose is marked for glycolysis. It can no longer escape the cell. This is done by the enzyme hexokinase in all the cells of the body, but in liver, this enzyme is a bit different and is called glucokinase. And obviously, you use ATP to add the phosphate. Next, glucose is converted to fructose using hexoisomerase. Next, fructose is charged again, fructose 6-phosphate, by the addition of another molecule of ATP by phosphofructokinase, and you have fructose 1,6-bisphosphate. Fructose is finally broken down into two different sub sugars, which is glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, by the enzyme aldolase. The excess dihydroxyacetone phosphate can take part in other uh, metabolism processes, but in for glycolysis, it is converted to glyceraldehyde 3-phosphate by phosphotriose isomerase. And here, the first addition of an extra phosphate is done using inorganic phosphate and NAD plus, where glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. And now production of ATP occurs by phosphoglycerate kinase, where one phosphate molecule is removed. You form 3-phosphoglycerate. This is changed to 2-phosphoglycerate and then phosphoenol pyruvate. And finally, another molecule of ATP is produced by pyruvate kinase. So essentially, you have put in two molecules of ATP in the charging process. And since you broke, fructose is a 6-molecule sugar, it is broken into two 3-molecule sugars where you add inorganic phosphate. So essentially, when you take ATP out, you get two in this step and two in this step. So in throughout this process, you get a net gain of 2 ATP. So this is how the entire process works. And regulation of glycolysis. So glycolysis normally goes on like this, but the cell can receive signals from the body which tells us that if there is requirement for a lot of glycolysis or if there is a lot of energy and there is no requirement for glycolysis, then we shouldn't keep on degrading the glucose. So that is done by controlling two steps in the entire process, or rather three steps, which are these three: the irreversible steps, hexokinase ATP addition, phosphofructokinase ATP addition, and pyruvate kinase ATP, where the ATP is taken out from phosphoenol pyruvate. These three steps are the ones that can be controlled. So what happens? So when there is low blood glucose level, so when there is a low blood glucose level, uh, which is on this side, the pyruvate kinase is phosphorylated, and you have less activity of the last step of glycolysis. So this step is hampered, and thus production of pyruvate is halted. So you do not take out the last ATP because you do not need it. You do not need the entire process to go through. But if there is more of glucose in the body, then you want more and more glycolysis to take place. So it is dephosphorylated, and then you have more production of ATP. So glycolysis goes on normally, and this is known as a feedback mechanism in which one of the later products in the entire chain inhibits the previous steps. So by controlling one of the later products, you inhibit or activate the entire process. This is also done, as I said, on the third step, which is conversion of fructose 6 phosphate to fructose 1,6 phosphate, and you do it by using so normally this step is done by phosphofructokinase, but there is another enzyme which is called for fructose 1,6 bisphosphatase, and this enzyme can inhibit phosphofructokinase because there is a lot of production of fructose 1,6 phosphate. So when the body has a lot of glucose going around, lot of glycolysis is forming happening, then even in addition of fructose 1,6 phosphate, you also form another byproduct, which is fructose 2,6 bisphosphate. When this keeps on accumulating in the cell gets to know, yes, there is a lot of glycolysis going on. That is why I have accumulation of a byproduct, and this byproduct actually inhibits phosphofructokinase as well as inhibits fructose 1,6 bisphosphate to let the reaction go in the opposite direction. So you have fructose 6, so essentially glycolysis halts at fructose 6 phosphate. And what is the total ATP balance of the entire glycolysis? During the priming step of glucose, steps 1 to 4 or 1 to 5, you have invested two glucose molecules, two ATP molecules. When then you generate two molecules of ATP from each glyceraldehyde 3 phosphate, because glucose is a 6 carbon sugar, and you have broken it down into two molecules of glyceraldehyde 3 phosphate. For each molecule of glyceraldehyde 3 phosphate, you are generating three molecules of ATP, which finally comes down to 6. And then NADH will give us one each NADH that is produced in the fifth step gives us one a 3 ATP. So that is also what you calculate, and then you finally generate two molecules of ATP. So finally, when you calculate, you get 8 net molecules of ATP when NADH oxidation takes place. That is the uh, oxidative phosphorylation that happens in the mitochondria is taking place normally. Then you produce 8 molecules of ATP from glycolysis. If this does not happen, then you only produce 5 molecules, because NADH from glycolysis itself, you only produce 5 molecules of net ATP increase. So low blood glucose level causes phosphorylation of pyruvate kinase. Next, in nucleic acids, blank provides the backbone to the polymer. The nitrogenous bases, the phosphoric acid, the ribose sugar, or the hydrogen bonds. Okay. So Shivakumar, Devadita, and Chandra all say D, phosphoric acid. So nucleic acid is the backbone for all the genetic material in all organisms. So they form a polymer known as deoxyribonucleic acid or DNA, and that contains the hereditary or genetic information in most organisms. In some viruses, RNA can play the same role. So DNA or RNA, it is an acidic molecule. That is why the name includes nucleic acid. And the first nucleic acid was isolated by Friedrich Miescher in 1868. What does one molecule of nucleic acid look like? There are three major components. One is the ribose sugar, which is essentially what holds the entire nucleic acid together. The ribose sugar, if it is a normal ribose sugar, it is just called ribonucleic acid, which is RNA. In case of DNA, the 2' hydro OH or the hydroxyl group is doesn't have so there is not a hydroxyl group but a hydrogen present at the 2' region. So it is known as deoxyribose or 2-deoxyribose, which is DNA. Then you have a phosphate group, which is attached to the 5' end of the ribose, and a nitrogenous base, which is present at the 1' end of the ribose. So this is what your nucleic acid looks like. One end you have a nitrogenous base. It could be your ATGC, the adenine guanine thymine or cytosine. In case of RNA, you do not have uh, thymine, but you have uracil, which is a separate nitrogenous base. Then you have your deoxyribose sugar, and then finally you have your phosphate. And how does the poly? So this is one nucleotide, and these nucleotides come together to form the entire huge polymer of DNA. And this polymer gets formed by by a esterification reaction of phosphodiester group forming between the phosphate and the 3' OH. So there will be a bond between this OH and this phosphate, and it looks something like this. So you have your 3' OH, which binds to the phosphate as in a phosphodiester bond, and this forms the backbone. So this 5', then you have the 3' OH forming another bond with the phosphate. So you have this phosphodiester backbone that links the entire DNA together, and the ribose and the bases stay on the inside of the DNA double helix. So in nucleic acids, blank provides the backbone to the polymer, the answer is phosphoric acid. Next, in Griffith's experiment, transformation was prevented by DNA's digestion, indicating that blank is responsible for carrying genetic information. RNA, protein, DNA, or lipids. Okay, Shivakumar, Chanda, and Devadita also see DNA. So, what is this experiment? So, very early, Frederick Griffith was the person who was experimenting on bacteria, and at that time there was still a lot of contradictions and arguments regarding what exactly is the genetic material in the cell. So, what exactly carries information from one cell to the next generation of cells? And people were still had this idea that it could be cytoplasmic factors such as proteins. And Frederick Griffith actually performed a lot of experiments in 1928 involving bacterial transformation that proved that DNA is the genetic material. So let us look at what the experiment was. So Griffith was working with two different strains of Streptococcus pneumoniae, which were S and R, the smooth strain, which was virulent, and the rough strain, which was avirulent. So both of these are the same bacteria, but they have something different between them that causes them to be either smooth or rough, and thus the smooth being virulent and the rough being not virulent. So, what was the experiment? So, the S3 strain or the smooth strain was virulent and possessed a lipopolysaccharide capsid and could kill mice by causing pneumonia and made round colonies on the culture plate. So the colonies looked roundish and it had a lipopolysaccharide coating helped it being which actually caused it to be virulent and cause pneumonia. But R2 strain was avirulent and lacked a lipopolysaccharide capsule and gave rough shaped colonies on the culture plate. So this is all the information he started with. So he designed an experiment. The first thing he did was he infected the mouse with the smooth strain and the mouse promptly died, which was something he knew before. Similarly, when infected with the rough strain, the mouse survived, which is also previous information. But here is where it becomes interesting. He hypothesized that whatever this information that is present in the smooth strain can be transferred to the rough strain. And what he did is that he heat killed the smooth strain. So the smooth strain, the bacteria are not alive anymore. And when injected, the mouse survived with this particular strain. But when this heat-killed strain was mixed with the rough strain of Streptococcus pneumonia, what happened is that whatever genetic material was present in the smooth strain was transferred to the rough strain, and when injected into the mouse, it caused the mouse to die. So essentially, there was transfer of genetic information from this heat-killed smooth strain to the rough strain. And when this strain was from the dead mouse was taken out, they found out that this particular so the when he injected a mixture of heat killed S and live R bacteria, the mice died, and the living S bacteria was recovered from the dead mice. So somehow the R strain had changed into the S strain. So there was obviously transfer of information between the two bacteria. Griffith concluded that the R strain bacteria had somehow been transformed by the heat-killed S strain bacteria. So some transforming principle transferred from the heat-killed S strain had enabled the R strain to synthesize the smooth polysaccharide code and become the S strain, and this must be due to the transfer of genetic material. But what exactly was the genetic material was not yet identified. This was done by Oswald Avery, Colin McLeod, and McCarthy, who took the experiment one step further. So, to determine which biochemicals from the heat killed S cells could convert live R cells into S cells, they isolated different biochemicals from the S cells, and they found that DNA from the S cell was all that was needed to convert the R bacteria. So essentially, essentially they took the heat killed S strain and the live R strain, but treated the S strain with protease. So this will destroy all the proteins in the heat killed S strain, but they still saw that the mouse died. So whatever information was traveled from the S strain to the R strain was not via protein. Similar, using RNAs, they showed that if RNA is removed, the mouse still dies, which shows that it was not RNA that carried the information from the S strain to the R strain. But when they treated it with DNAs, then the mouse was live, and then they couldn't find any S or smooth type bacteria. Thus, DNA is the information that was traveling from the smooth strain to the live strain, and this use of DNAs prevented this genetic information transfer and caused the cell mouse to live, and thus DNA is the genetic material of the cell. So what the conclusion they were brought to is they found that both RNA and protein digesting enzymes had no effect on transformation, proving that molecules undergoing transformation was neither a protein or a RNA, but the transformation was prevented by DNA's digestion, indicating that DNA was the transformation cause. And they finally came to the conclusion that DNA is the genetic material in the cell. But there was still some argument that the further experiments put down. So, but for this question, in Griffith's experiment, transformation was prevented by DNA's ligation, indicating that blank is responsible for carrying information. The answer is DNA. Okay, next. The sequence-dependent synthesis of DNA from pre-existing DNA is known as transcription, translation, replication, or repair. Okay, Shivkumar and Chanda both have said it is C, which is replication. So just look at it. So the key central idea towards molecular biology is the central dogma of life, as postulated by Watson, as postulated by Crick, rather, which is the flow of information occurs from DNA to RNA to protein via different mechanisms, but essentially DNA is the genetic material of the cell, which is transcribed to RNA, and RNA is then translated to proteins, and the proteins are the actual building blocks of life. So this is known as the central dogma of life. And there are various different steps that he had proposed. First is replication of DNA. So DNA dependent DNA formation done by a DNA polymerase is known as replication. Then there you have DNA dependent RNA synthesis, which is done by our RNA polymerases, which is DNA to RNA. And finally, translation, which is RNA dependent protein synthesis, which is done by ribosome. So this is essentially the central dogma as it was first proposed. DNA to RNA to proteins, and only DNA can form more DNA. There is no backtracking of information from this part. So from proteins to DNA. But there were some caveats that were later added where people figured out there's an enzyme which is called RNA transcriptase, which can form DNA from RNA, which are present in some viruses. And RNA transcriptase is a RNA-dependent DNA polymerase, so this step can be reversed. But this information is still known as the central dogma of life, and this is key to all molecular biology. So, what is replication? Replication is the genomic DNA content in an organism needs to be duplicated during S phase of the cell cycle. Duplication of DNA is done by replication utilizing the sequence information of the parent DNA, and the enzyme used for this purpose is the DNA-dependent DNA polymerase. Next, transcription. The DNA is present in nucleus, whereas protein synthesis machinery is present in the cytosol. Hence, the information present in DNA is used to synthesize RNA, which has the ability to transport outside the nucleus and participate in protein synthesis. Synthesis of RNA from DNA is done by transcription, utilizing sequence information of DNA. The enzyme used for this purpose is DNA-dependent RNA polymerase. And finally, translation. The RNA presenting cytosol is used by the translational machinery to synthesize proteins in a sequence-dependent manner, which is translation. So DNA to DNA replication, DNA to RNA transcription, RNA to protein is translation. So the sequence-dependent synthesis of DNA from pre-existing DNA is known as replication. Next, telomerase adds telomeric repeats of the parental DNA ends using blank as template for synthesis. Is it a DNA component? Is it telomeric RNA, self RNA component, or ribosomal RNA? Okay, we have our answers coming in. Hmm. Shivakumar Devadita says it is B, telomeric RNA, and Chanda says it is C, self RNA complement. So let us look at it. So what are telomeres? So telomeres are this uh, repeat DNA sequences present at the end of the chromosomes. These are short, these are have tandem repeats, that is, these repeats repeats, and these are GCA-rich nucleotide sequences. Why are telomeres actually essential? So during eukaryotic replication, telomeres play a crucial role in ensuring the accurate replication of linear chromosomes. Eukaryotic cells use a semi-conservative mechanisms to duplicate their DNA, and this process poses a challenge at the ends of linear chromosomes, and telomeres overcome this challenge by maintaining the integrity of genetic information. So why does this happen? So we have discussed DNA replication before. So there are whenever there is a replication bubble, so that is the two strands separate and DNA synthesis occurs, there is always a leading strand, so where the replication is taking place from the 5 prime to the 3 prime direction, which is where the DNA polymerase moves. But there is also the lagging strand, which is the opposite strand, and obviously the replication is now occurring 5 prime to 3 prime, but away from the replication fork. So imagine we imagine it like this, that for the leading strand, as the replication fork keeps on opening, the DNA polymerase can keep on just adding and adding and adding and polymerizing the new new leading strand. But for the lagging strand, the moment DNA polymerase sits somewhere and forms a segment of the lagging strand, the replication fork has gone ahead in the opposite direction. So there obviously will be gaps because it is only going to be able to synthesize in the 5 prime to 3 prime direction. So the synthesis of the lagging strands actually occurs in fragments, which are known as Okazaki fragments. So once the replication fork moves ahead, there will be made a RNA primer by the enzyme primase, and DNA polymers will sit and synthesize a small fragment of the lagging strand, which is then ligated to the rest. As the replication part moves ahead, another small RNA primer will be made, and that part will be formed. So essentially, the lagging strand is made in these bits and pieces, whereas the leading strand is made in one go, essentially, with there being a long leading strand and the lagging strand being formed in Okazaki fragments. And obviously, you also probably remember that DNA polymerase, even though it is a DNA dependent DNA polymerase, it cannot initiate polymerization without a primer, this short stretch of already double-stranded DNA. So this primer is supplied by the enzyme primase, and it is a RNA primer. So this is where all the problems come in, which is known as the end replication problem. So if we know that there is going to be a primer to be formed at the lagging strand, so when you reach the end, then when the primer, so at the end, if you put in a primer and that gets formed and the primer is then removed, then you have a gap present which is not replicated into the new strand, because this is the place where the primer sat. And without there being something behind it, the DNA polymerase cannot synthesize this region only. So there would always be a end replication problem. So linear genome, including those of several viruses, as well as chromosomes of the eukaryotic cells, pose a special problem in completion of the replication of the lagging strand. Excision of the RNA primer from the 5' end of a linear molecule would leave a gap known as the primer gap. This primer gap cannot be filled by action of DNA polymerase because of the absence of a primer terminus to extend. If DNA could not be replicated, the chromosome would shorten with each round of replication, and thus you will have a loss of genetic information. And but this problem was solved by the enzyme which is known as telomerase. So what is telomerase? Telomerase is a RNA-dependent DNA polymerase. It is a ribonucleotide protein RNA component having repeats of 9 to 30 nucleotides long. This RNA component serves as the template for synthesis of telomeric ends. Hello. Okay. Does anyone have any questions? Someone has their mic unmuted. Hello. Ashini, if you have any question, then you can put it into the chat. Yes. Coming back. So it is. So what is telomerase? Telomerase is a ribonucleoprotein containing an RNA component having a repeat of 9 to 30 nucleotides long. So the protein itself, however, has an RNA component embedded, which is 9 to 30 nucleotides long. And this RNA component serves as a template for synthesis of telomeric repeats at the parental DNA end. So what telomerase does is it makes DNA based on the template that is already embedded in it. So the 3' of the parental DNA strand acts as a primer, and the template is the self-RNA component. And what is the function? It has a 5' to 3' RNA-dependent DNA polymerase activity, due to which it adds successive telomeric ends to the parental DNA strand at its 3' end. So at the end where the primer synthesis has stopped, and so the when the DNA polymerization normal has stopped because there is a primer gap, the telomerase comes in and uses its own RNA component as the template to keep on adding bases to the end of the DNA. So telomerase adds telomeric repeats at the parental DNA ends using the self RNA component as template for synthesis. Okay, next. When lactose is not available, the lac repressor blank with the operator of the lac operon. Weakly binds, strongly binds, dissociates, or loosely associates. Okay, so Chanda and Devadita both say B, strongly associates. You just look at it. So we have learned about the lac operon previously. So lac operon has essentially so lac operon is made is this huge operon made of the promoter, then an operator where a different protein can bind, and the functional genes like Z, Y, and A, and a cap binding region. The operator can be bound by another the product of another gene called LAC-I or LAC inhibitor, and the cap can be bound by CAMP binding protein, which binds to CAMP and then binds to the cap binding region. So how does the lac operon function? So when So lack operon. So one thing we have to understand is that lack operon, the genes in the lack operon should only be transcribed when there is lactose present but no glucose present, because glucose can be much more easily metabolized than lactose. So there is no need for lactoferol to be transcribed and the genes for lactose metabolism to be transcribed, which takes a lot of transcribed and translated, which just takes a lot of energy when there is a lot of glucose present. So when the glucose is present, the lactoferol should stay closed, but when glucose is not present and only lactose is present, then it should start translating the lactose metabolism genes in high quantities. So let us look at what different conditions happen with the lack of operon. First, when the glucose is absent and the lactose is present, when we mostly need our lactose metabolism genes to be translated. So strongest transcription of lac operon occurs in this case. The lac repressor, which is the lac I gene product, cannot bind to the operator because of the inducer, which is allolactose, is present. So this is this image, the image B. So here is where the lac inhibitor has an allosteric site for allolactose. Allolactose is a byproduct of lactose once it starts entering the cell. So the moment allolactose is present means there is lactose, and that prevents the lac inhibitor from binding to the operator region. So essentially, the operator is turned on. Next, CAMP, which is the hunger signal of the cell, which says that this lactose is present, which inhibits lac inhibitor, and CAMP tells that the cell does not have any other source of energy. So this causes C so CAMP levels are high because glucose is absent, and thus CAMP activates CAP and helps binding of RNA polymerase to the promoter and allows for high levels of transcription. So this is the best case scenario for lactose synthesis when the lactose metabolism, when lactose is present but glucose is absent, and this is when the strongest transcription happens. But what happens when glucose is absent as well as lactose is absent? No transcription should occur from the lactoferol. So even though CAMP levels are high and cap binding protein has bind to cap region and recruited the RNA polymerase, the lac inhibitor is still sitting on the operator region of the lactoferol and prevent the RNA polymerase to transcribe the downstream genes. The lac repressor will also be bound to the operator because there is no inducer, which is allolactose, because there is no lactose, and acting as a road blocker to the RNA polymerase and prevent transcription. So no transcription in case there is no lactose and low glucose present. Next is glucose present and lactose absent. In this case, low level of transcription of the lac operant occurs. So when lactose is present, we have allolactose again, preventing the lac repressor to binding to the lac operator. But the lac repressor cannot bind to the operator because the inducer allolactose is present. But because there is glucose also, this causes low levels of CAMP. Once if CAMP is not there, cap will not bind to the cap binding region, and thus RNA polymerase will not be strongly recruited to the lac operon. So there will be a low level of leaky transcription from the operon. And finally, glucose present and lactose absent. Again, no transcription will occur because the lac repressor is free to bind to the operator because there is no lactose. So lac repressor remains bound to the operator and prevents transcription by RNA polymerase. Further, CAMP levels are low because glucose levels are high. So cap does not bind to the cap binding region and RNA polymerase is not recruited here. So there will be no transcription at all from this lac operon. So to summarize, if glucose is present and lactose is absent, cap binding does not occur because there is no starvation in the cells and the repressor binds too, so no transcription. Glucose is present, lactose is present. In this case, repressor will not bind, but cap also will not bind. So low level of transcription, glucose is absent, lactose is absent, cap binds to the lac operon, but lac repressor also binds, preventing transcription, so no transcription. And finally, glucose is absent, which increases the levels of CAMP. Lactose is present, which removes the repressor, and CAMP levels allow cap to bind, so there will be high levels of transcription. So when lactose is not available, lac repressor strongly binds to the operator of the lac operon. Any questions from this? Okay. The code blank is blank, which suggests that each codon specifies only one amino acid. So this is talking about the amino acid code. So the amino acid code is blank, which suggests that each codon specifies. Chanda says, why is the lac operon inducible? So so whenever we talk about an inducible operon, it means that the operon is normally off. So when we saw this, we saw that till then you add lactose, the normal condition is when the lac repressor will bind to the operator and prevent any transcription. So it is normally off, and you can induce it. You can external factors have to turn it on. So that is called a inducible operon. Further, we have also figured out that instead of using lactose, which will be then used by the cell in order to be metabolized, and then thus the amount of lactose you add will decrease over time, you can actually use the synthetic analog of lactose, such as IPTG, which will cause the lac operon to be active because it will remove the lac repressor, but it will not be catabolized by the cell. So IPTG small amount can keep on inducing the lac operon, and you will always have high levels of transcription from the lac operon. But why is it inducible? Because normally it is off, and you can turn it on. The other uh, the other type of operon is where you it is normally always transcribing, but you turn it off. So that is why lac operon is called inducible. Is that fine? Okay. Next, the amino acid code is blank, which suggests that each codon specifies only one amino acid. The options are degenerate, universal, unambiguous, or redundant. Okay, we have multiple answers. Devagita and Chandra both said it's C. Unambiguous. You just look at it. So the genetic code that codes for the amino acids, which is important for translation, is a triplet code in nature. So three nucleic acids code for one amino acid, and this is known as a codon. So the genetic code is triplet, and it is known as a codon. Each coding sequence has a start and a stop codon to initiate and terminate translations respectively. The start codon is usually AUG, which codes for methionine, and the stop codons could be UAA, UAG, or GGA. The code is unambiguous. What unambiguous means is that the same three letters of the nucleic acids will always code for the same amino acid. There is no confusion that CUU will ever code leucine or not. It will always code for leucine. So the code is unambiguous, which suggests that the code is for only one amino acid. The code has no comma or gap. It is continuous. The code is degenerate, which means that one amino acid has more than one codon, which you can see right here. Leucine has four. So it is degenerate that the same different sequences of nucleic acids can code for the same amino acid, but it is unambiguous. Once that code is present in the nucleic acids, they will always synthesize the same amino acid. So both ways. So, for example, alanine is specific to two codons UUU and UUC. Only tryptophan and methionine are coded by a single codon. Next, the code is non-overlapping. For a code, for example, starting with AUG, CUG, GGU, then the codons will be AUG, CUG, GGU, and so on. And not you take these three and then you start somewhere here or somewhere here. You take these three and then you just shift to the next three. So it is not non-overlapping. So it is sets of three, one after the other. The genetic code is universal, which suggests that the genetic code and its meaning is common for all life forms. But there are some exceptions. For example, UGA is a stop codon, but it codes for tryptophan in mycoplasma, spiroplasma, and mitochondria of several species. Similarly, CUG for leucine in general, but in yeast code, mitochondria it codes for threonine. But it is genetic code, we take it as universal. So the genetic code is unambiguous, which suggests that each codon specifies only one amino acid. The ATS ribosome dissociates to form blank subunits. Shivkumar and Chandra both say it is C, 40S and 60S. So just look at it. So now we are discussing translation. So translation has different. So translation is done by the translation machinery. The most important of which is the ribosome, and the major step in ribosomal machinery taking part in translation is elongation of the peptide that is getting formed. So it occurs in three different steps. First is decoding. It is the codon is directed towards the binding site of the ribosome, and during this process, the ribosome selects and binds to the incoming amino acyl tRNA at a site whose anticodon is complementary to the codon of the mRNA. And the decoding region of the 16S RNA confirms the proper base pairing between the codon and the anticodon. Formation of the peptide bond. In this process, the peptide group of the P site of the tRNA is transferred to the amino acid group inner site through the peptide bond, and translocation. In this case, tRNA of the A site is transferred to the P site to make a space for the next amino acid tRNA at A site, and the A site of the tRNA is shifted to the E site. So this sounds confusing, but this is essentially the A site, P site, and the E site. A site is where the amino acid tRNAs are coming in place. P site is where the peptide bond is getting formed, and E site is where the exhausted tRNA is getting removed from the or exits from the ribosome. So first, the so normally the P site has the growing peptide chain. There is a charged tRNA which contains anticodon that is complementary to the codon of the mRNA, and on the opposite end carries the corresponding amino acid. It comes in the A site. The tRNA is matched to the codon on the mRNA, and the ribosome confirms that yes, this is the codon here. Then the P site peptide bond is moved to the A site, and thus the peptide grows one site longer. The tRNA that was being that was right here moves to the exit. Then the ribosome moves one step forward. The tRNA that just came in, which now has the longer peptide chain attached to it, moves to the P site. The exhausted tRNA exits via the E site, and a new tRNA comes in the A site. So essentially, the chain keeps on getting longer, and it keeps getting on passed between the P site and the A site. So here again, this is where the new tRNA comes in. The pair codon and the anticodon of the tRNA match. The amino acid chain from the P site. So this part will transfer here. So this tRNA will have one the amino acid longer now attached to it. This tRNA, which has its amino acid removed, moves to the exit site and goes out. This tRNA, which now has a longer chain, moves to the P site. The entire ribosome moves forward, and a new tRNA comes in. And how does the termination take place? One of the stop or termination sequence, UAA, UAG, or GGA, terminates the growing polypeptide. When the ribosome enters stop codon, there is no tRNA available to bind to the A site of the ribosome. Instead, a release factor binds to it. ERF1 recognizes all three stop codons, and ERF3 stimulates the termination events. Once the release factors bind the ribosome unit falls apart, releasing the large and the small subunits. The tRNA carrying the polypeptide is also released, freeing up the polypeptide product, and ribosome recycling occurs at the end. And in case of eukaryotes, which have a 80S ribosome, it divides into a 60S and a 40S ribosome. The 60S is the largest subunit, 40S is the smaller subunit. In case of eukaryotes, it is a 50S subunit and a 30S subunit. So the 80S subunit dissociates to form the 40 and the 60S subunits of the ribosome. Any questions? Yes, Chandra, go ahead.
Why the non-template strand is coding strand?
Why is the non-template the strand known as the coding strand? Okay, for that we will have to go back a bit. This is rather a question for not a transcription. Do I have a slide on transcription? No, I don't. So, okay, I will tell you why. So, when you have transcription happening, so you have two strands of DNA. One strand is the top strand, one strand is the bottom strand. So, the top strand, when our RNA polymerase sits on the top strand and starts transcribing the mRNA that will be transcribed is complementary to the top strand. So say you have ATGC as the sequence of the top strand, then whatever the mRNA will form will be complementary to that. So it will be TACG, which is complementary to the top strand. Now, whatever the mRNA is getting formed, that sequence is complementary to the top strand. But what else is complementary to the top strand? The bottom strand. So the sequence of the mRNA will be exactly same to the sequence of the bottom strand. And that is why when we talk about transcription, we have one strand known as the template strand from which actual mRNA is getting formed. And one other strand is known as the sense strand, which has the same sequence as the mRNA. Whereas the template strand has the complementary sequence. So if you ever want to figure out the sequence information of a particular which like what will the mRNA look when I transcribe this particular strand, then it you just have to look on the other strand that the sequence will be same. That is why the other strand is known as the sense strand or the coding strand, and the bottom strand is known as the template strand from which the mRNA will get made, or the nonsense strand, or if there are five more other names. But essentially that is the idea. Is that fine, Chanda?
Yes, sir.
Okay.
Sir, I have another question.
Yes, go ahead.
Why protein does not directly synthesize in DNA?
Why protein is not directly synthesized from DNA is what you mean. So the reason there could be multiple reasons, but that one could be that evolution has just done it that way. One other reason that we can say is that DNA is where you have the genetic information being stored. You want that to be as less touched by everything else as possible. But if you start translating RNA, protein directly from DNA, then there will be 50 different ribosomal machinery, and various different um proteins that have to access the DNA that can cause DNA damage. And then you will start losing genetic information. It will be better if you have a blueprint, and from blueprint you make copies, which are the RNA, and using the RNA you make stuff. Because then your original blueprint does not get destroyed. So thus you have a DNA, you then have a RNA on which you everything can go ahead and start translating. You have splicing, you have 5' capping. You can control the amount of expression of the protein by the different post-transcriptional modifications on the RNA. But all of this will not hamper your DNA. The information is still there. The information is untouched by all these enzymes, but you are still able to make it because make the protein because you are using a middleman in between. But yeah, so their ribosomes essentially cannot read the DNA directly. They can read the RNA, and that is how just how we have cell and molecular biology has developed. Is that fine?
Yes, sir. I have last one question.
Yes, go ahead.
Can ligase can be used as polymerase?
And ligase cannot be used as a polymerase because the action of a ligase is very different from that of a polymerase. Ligase is only useful for joining together two phosphodiester, so a broken phosphodiester bond between a 5' OH and a 3' phosphate. It will just join it together. DNA polymerase, what it does is that it forms that phosphodiester bond but adds a new nucleotide at its end, depending on whatever template it is working for. For example, during DNA replication, it will keep on forming those phosphodiester bond as it reads the template and adds more and more nucleic acids corresponding to that template. But a ligase does not read anything. It just looks for a broken phosphodiester bond and joins it together. Thus, there has to be a phosphodiester bond that is broken. So it cannot make something but repair. The DNA polymerase is what makes it. Another interesting question could be, can a DNA polymerase actually act as a RNA polymerase? Can the same enzyme do both transcription as well as replication? and they currently there has been a lot of research being done in the area, and people have been trying to make a DNA polymerase can also do transcription work as a RNA polymerase as well. Anything else?
No, sir. Thank you.
Okay. Okay, going on to the next question. In southern blotting, the DNA fragments are separated by size using blank. The options are high performance liquid chromatography, capillary electrophoresis, agarose gel electrophoresis, or SDS page. Okay, Chandra and Shivkumar both say it is C, which is agarose gel electrophoresis. Both of you are correct. So let us look at it. So separation of DNA on agarose gel. So agarose gel electrophoresis is a standard gel to resolve DNA. As a practice, ultra pure quality of agarose is used. So agarose is this protein isolated from seaweed, and this protein, when you mix agarose in water, it is normally does not dissolve, but you heat it up and let it cool, then it will start forming cross links and will form this jelly-like structure, which we call the agarose gel, and it is very uniform in nature. So all of the pore sizes of the agarose crosslinks are very uniform, which allow it to be a very powerful tool to separate out biomolecules, especially DNA. You put in your DNA and then you apply a electric field across it. So we know that DNA is negatively charged. We'll try to move towards the positive electrode. So you add a positive electrode at one end, negative on another end, and the DNA will move from the negative to the positive electrodes depending on how big the DNA is. Because DNA is also uniformly charged, 10 bases will move much faster than 100 bases. And thus, depending on the weight of the DNA, you are able to separate it out. And it is known as agarose gel electrophoresis. And this is here, and then you can also load a ladder. So ladder is a pre-digested set of DNA bands which tell you what the size is. So this is a 1 kb ladder. If you are able to read it, you will be able to say that this is say 3 kb, this is 2 kb, and thus this band in between must be 2.5 kb around. And so all the bands on the gel is 2.5. So this is the you know what the size of the ladder would be, what the size of the different bands on the ladder would be, and comparing it to how much your DNA of interest has traveled, you can specify how big your DNA was. And this is what the final resultant gel looks like. And you, in order to visualize DNA, because obviously you can't see DNA with your naked eye, you use fluorescent markers such as ECGM bromide, which intercalates, which goes inside the double helix, and then once it goes in, it can start fluorescing in UV. And you throw UV light at the gel, and the DNA will glow as bands such as this, and then you compare that to your ladder, and you will be able to see, yes, this band is of 10 kb, this is of 11 kb, and whatever, and everything else. So in southern blotting, the DNA fragments are separated by size using agarose gel electrophoresis. Next, blank is used for the preparation of RNase free water. The options are DEPC, DMSO, ethanol, or formaldehyde. Farhan, if you have any questions, then you can put it in the chat directly. Okay. Chanda says it is A, depth C, which is used in preparation of RNA-free water. So whenever you are working with RNA, you have to understand that RNA is extremely labile in nature. Even heating it up will cause RNA to denature. And further, there are various different RNases, enzymes which chop up and degrade RNA going around everywhere. So you have to prevent your RNA, if you are working with RNA, to get degraded. So you use a compound known as DEPC. DEPC looks like this, and what it does is that whenever you mix it in with any enzyme or polar group, it will form a bond with them and cause the particular so it actively forms bonds with various different polar amino acids. And once it does, it actually causes them to misfold and it essentially inactivates various enzymes. And the advantage of using DEPC is you can be able to you will be able to denature any RNAs that are going so any RNAs that are going around in your water and thus or buffers, and thus you are able to use them for your experiments. So blank is used for the preparation of RNA-free water. This is step C. Muhammad Quran is asking exam question. So, Farhan, unfortunately, I am not the person who is designing the NPTEL exam questions. That will be done by the PI who is taking the course. So, I do not have access to the questions. I'm just going through the ones that I find will be helpful for you to go through. But I do not have any idea what the questions in the final exams are going to be. Okay, the function of SSB protein is blank. Prevent re-annealing of single strands during a DNA replication, increase DNA polymerase activity, prevent DNA denaturation, assist in primer synthesis. So Shivakumar says it is A, prevent re-annealing of single strands during DNA replication. Just look at it. We know that the DNA replication, there are three steps. To initiation, where DNA is bound by different proteins and the double helix is separated to initiate the entire DNA replication process. Elongation, where the synthesis of the new strand is done by DNA polymerase, and termination, where the DNA synthesis stops either by the row or dependent or row independent mechanism. And there are different proteins that play a role in the entire process. The first is helicase that separates the two strands. Primase forms the RNA primer that the DNA polymerase is used for DNA replication. SSB or single strand binding proteins. So once the helicase separates the two strands, these single-stranded binding proteins bind to the single strands and prevent them from coming together again to allow the other proteins to come in and do the synthesis. So it prevents the re-annealing of the single strands. DNA polymerase obviously synthesizes the new strand, and tethering protein stabilizes the polymerase to the DNA strand. And yeah, look at this one. Function of SSB protein prevent re-annealing of single strands during DNA replication. Next, real-time PCR is also known as blank, conventional PCR, reverse transcription PCR, sequencing PCR, or quantitative PCR. Chandan Shivkumar both said D, which is quantitative PCR. Let's just look at it. So, what is real-time polymerase chain reaction? So, the real-time PCR is a combination of both amplification of target DNA using PCR, but also having something else in there to let you know how much DNA is getting amplified over time. So the real-time PCR combines Amplification of gene of interest with intensity of fluorescence. This can be done using Tachman probe or cybergreen method that we have discussed previously. The moment at which the target application is first observed mainly defines the reaction. The time period at which fluorescence intensity exceeds the background fluorescence intensity, it is called cycle threshold or CT value. As a result, a large amount of target DNA, the fluorescence signal will appear more quickly, resulting in lower CT values. And the other name for real-time PCR is quantitative PCR. So this is how real-time PCR works. You have a template, you denature the template, and you have your forward and reverse primer binding to it, and then your DNA polymerase will synthesize the strands where the primers are essentially doing a normal PCR. But in case of real-time PCR, you have a Pacman probe bound somewhere downstream of this primer, and the Pacman probe is made out of a short DNA strand, a quencher, and a fluorescent reporter. The fluorescent reporter can fluoresce if it is away from the quencher, but in the Pacman probe, it does not fluoresce. Once the polymerase starts making the new strand, it actually bulldozes through and breaks up this Tachman probe, causing the fluorophore to escape. And once it escapes away from the quencher, it starts releasing a fluorescent signal, and thus you have increase in fluorescence as more and more of your DNA uh, is getting synthesized. So you will see the sigmoidal curve, which shows the amplification, and thus you will be able to see how much of your DNA is getting made in real time. So real-time PCR is also known as quantitative PCR. Next, homopolymer tailing exploits the presence of blank present on mRNA to synthesize the first DNA strand. 5' cap, poly A tail, poly G tail, or splice sites. Okay, both of them are saying Shivkumar and Chandra say B, poly A tailing. So just reminding us of the mRNA structure of mRNA in eukaryotes. You have a 5' cap, which prevents exonucleus activity from different RNases, a 5' UTR, where ribosome will be there but it will not translate. Then you will have your start codon, and where the ribosome will start assembling your peptide. The coding sequence that will be translated, a stop codon where ribosome will fall off, 3' UTR which the ribosome will never see, and that is why it is known as an untranslated region, and a poly A tail. This is again used for protection of the RNA from exonucleases, because anything that starts chopping up, you'll have to chop up a lot of just random adenines, and it also helps in translation. But this poly A tail has been utilized by us to isolate mRNA. So what we do is that we homogenize our tissue from or cells from which we want to isolate the mRNA, and use oligo-DT beads. So oligo-DT beads, these are beads that have long uh, thymine tails, and this thymine will obviously base pair with our adenine poly A tail that is present in our mRNA. And then if we use these beads and then wash everything else off, we will be able to collect pure poly A RNA, which will be our mRNA of interest. And once you have your poly A RNA with you, you can use it to make a complementary DNA or cDNA because, as we have been saying, RNA is extremely labile. You don't want to store a RNA library. It will be preferable if you can somehow convert it to a DNA library, and we obviously have the mechanism to do it using a reverse transcription. So we always save, we convert, make the mRNA, and then like isolate the mRNA and convert into a cDNA library to store. So there are three steps: synthesis of the first strand using reverse transcription, a reverse transcriptase, removal of the RNA template, and second strand synthesis. So what you do, you take your mRNA of interest, which has a poly A tail, you put in a DT primer, and then allow it for reverse transcriptase to work, and you have a cDNA made. This is the first strand. Then after the cDNA is made, you can either use the alkaline sucrose gradient or RNase H to destroy the mRNA so that you only have the cDNA now. And once you have the cDNA at the end, you use an enzyme known as a terminal transferase to add cytosines to the other end. So you have the cDNA with T's at one end and C's at the other end, and then you use a DG primer, so a lot of G's at one end to prime the C's at the end of the cDNA and make the second strand using DNA polymerase PCR, whatever you want. And thus you have created a double-stranded cDNA from your mRNA, and this can be stored in your cDNA library. So homopolymer tailing exploits the presence of blank present on mRNA to synthesize the first strand, and it is poly A tail. Next, transposons are segments of DNA that can move from one locus to a relatively non-specific site in the genome and are known as A. jumping genes, B. intron sequences, C. exons, or D. regulatory elements. Shiv Kumar and Chandra both say it is A. jumping genes. So what are transposons? So transposons are segments of the DNA that move from one locus to a relatively non-specific site on the genome. The movement of transposons can occur with duplication, which is known as copy and paste mechanism, or they can just move from one place to the other, so it doesn't stay in the original place anymore, and it is known as the cut and paste mechanism. DNA transposes carry so DNA transposes interestingly the entire gene, the transposable element actually carries inverted repeats that help it integrate it anywhere in the genome, as well as codes for the transposon enzyme, which causes the cleavage of the transposable element out of its original location and integration into another site. So DNA transposes carry inverted repeat sequences at the end as recombination sites flanking a recombinase enzyme known as transposase, which bring about the process of recombination. Retrotransposons, which are viral transposons, contain LTR and genes for integrase and reverse transcriptase activity, which is the copy and paste mechanism, which is done by some viruses. So there are two methods, copy and paste, cut and paste. In case of cut and paste, we have DNA transposons. It makes a transposase enzyme, which cuts the gene itself and then integrates it somewhere else in the DNA. So cut and paste mechanism because it is no longer in the original location. The other is the copy and paste mechanism, in which you have viral retrotransposons. So they actually transcribe a mRNA, and the mRNA now is able to reverse transcribe itself into a double-stranded DNA and integrate somewhere else. So it is a copy and paste mechanism. What are the outcomes of the transposition? Altered gene expression through insertion within genes. For example, the IS1 element can insert itself near a AT-rich promoter sequence. IS elements can cause polar mutations, reg-dependent recombination, site-specific recombination between transposable elements in the same molecule can cause rearrangements such as deletions or inversion, etc. IS1 elements can cause deletion, TN3 and Y delta resolvase can cause resolvase reaction to deletion, and it can essentially causes mutations and frame shift mutations in general. So they are called jumping genes. DNA fingerprinting uses blank loci because these loci are so variable that unrelated individuals are unlikely to have the same VNTR. So the answers are options are where the DNA fingerprinting uses what loci? Variable number tandem repeat, single nucleotide polymorphism, short tandem repeats or restriction fragment length polymorphism. Shiv-Kumar and Chanda both say A, variable number tandem repeats or VNTR. So let us look at it. So there are various different applications of PCRs that we have, and one of them be DNA fingerprinting. It was previously DNA fingerprinting has been around for a while. Previously, we used restriction fragment uh, RFLP or restriction fragment length polymorphisms to do DNA fingerprinting, but nowadays we just use PCRs. So there are these particular genetic loci present in our DNA which have short tandem repeats of sequences, and these are extremely variable between individuals. So if you find DNA evidence on a crime scene and run a PCR against this VNTR region, then the length of the VNTR regions will be unique to the person who is probably was around the crime scene, and it is uh, extremely unlikely it is common between two separate individuals. It could be inherited between very close family, but mostly you can identify whoever the person was from that particular sequence. So each individual has a different DNA profile, which is known as DNA fingerprinting. A DNA fingerprinting uses variable number of tandem repeat regions or VNTR loci, as these loci are so variable that unrelated individuals are unlikely to have the same VNTR. A sample of blood, hair root, or tissue left in the crime scene can be used to identify a person using PCR. By comparing the DNA of the crime scene with that of the suspect with the DNA database or earlier convicts, evidence from decades-old crimes can be tested, confirming or defending the people originally convicted. So if you have a criminal scene DNA, you run the VLTR PCR for a lot of them, you find the one that matches the best, and that person is probably your suspect. So DNA fingerprinted uses blank loci because these loci are so variable that that unrelated individuals are unlikely to have the same VNTR, and the answer is variable number tandem repeats. Okay, so that's all I had for you today, and that brings our entire PMRF-NPTEL sessions for molecular biology to a close. You can find all the recorded videos at my YouTube channel in the molecular biology playlist, and if you have any questions that you want to come back to, you can always post them in the comments in the videos, and I will happily reply to them. So, does anyone have any questions to end us our session today?
No, sir. Thank you.
Okay. Thanks a lot to you as well for attending so diligently throughout the course, and best of luck for your NPTEL exam. Thank you.